welcome to this machine learning course for biologists. And today I'm going to introduce a little bit about machine learning fundamentals. So you can have this introduction before diving deeper into more technical topics in this course. So I think the best way to start is to define exactly what is machine learning. It's actually a combination, a mixture of different areas of expertise. I think you are probably starting more from here with your background, more focused on life sciences. You probably had some statistics, so you're applying that to your biological problems or to analyze your experimental data. And if you are more brave and you're already running some command line terminal tools like transcriptomic data analysis tools or microbiome data analysis or data preprocessing in the Unix or Linux terminal, you are developing some computer science skills to apply in your computational biology or bioinformatics tasks. And if you start combining math and statistics with computer science and developing machine learning models that can learn patterns from the data instead of establishing fixed rules like traditional statistics. So if you start to use algorithms and math to start learning patterns from your data, then you are starting to dive into the machine learning field. Combining all that, your life sciences background, computer science background, and math and statistics background, it gives you a nice set of skills that makes you become a data scientist for biology. And so I would define that my background is more or less like that. I have developed throughout my experience, throughout my career, throughout my foundational training, I have developed skills in each one of those areas. And I would define myself as a data scientist, for example. So the objective of this course is to introduce you to some computer science, some algorithms, some statistics and math to help you developing these machine learning skills so you can start to figure out how these techniques and how these machine learning methods can be used on your own data to extract new insights, more focused towards data-driven techniques rather than hypothesis-driven techniques to allow you to use these tools to learn new patterns and extract new insights from the data you might be generating during your undergrad or grad research throughout your PhD or master's, or even leveraging, revisiting publicly available data and applying these new techniques to extract new insights. So I would say this is a basic overview of what machine learning is basically leveraging computers to learn patterns from the data instead of setting fixed rules. And if we dive deeper into that a little bit, for example, if you are designing experiment and extracting data, microbiome, for example, if you use traditional statistics, you would have to set rules about how the data distribution looks like, about what exactly you expect to see in your data, set very specific thresholds, expert-derived thresholds that would define your data as a case or control, for example, or you would have to already know the rules that define your data. But in biological sciences, that's not true most of the time. Most of the time, we're generating new data that has been unexplored. We are in uncharted territory, and we don't know the rules that define a biological system. So how do we come up with those rules? Well, machine learning is a tool that can help you to come up with those rules. It can be hypothesis-free and dive into your data. You can leverage machine learning to actually discover what are the rules that drive the system, that define this complex system. Once you know those rules, then you can make new predictions. So why do we care about machine learning then? Why it would be useful, or why it would be even necessary? Like, can I get away just with traditional statistics, setting my own rules in a small system, or do I need machine learning? So there are situations where machine learning and AI and other data-driven techniques like that becomes a necessity. They become a need. And an example is when the biological data is too large and too complex for a human to understand just by looking at a spreadsheet. For example, with this next generation sequencing, transcriptomics, single cell sequencing techniques, sometimes we end up having spreadsheets with thousands of columns, with thousands of rows. And all those values are either integers or floating point values that make no sense to the human eye. So the data needs to be preprocessed, and it needs to run through machine learning models to identify meaningful patterns that are hidden within this huge amount of data that is overwhelming sometimes. One example are the gut microbiome studies, where people can extract microbiome compositions, gene expression levels, taxonomic information on the microbes present in the gut, the metabolites that they are producing, or even imaging data. So you have this highly dimensional data, you have a lot of features, a lot of different predictors, thousands of them, and you have hundreds or thousands of samples, and it becomes a problem that is infeasible to be solved manually. So that's where machine learning comes in, and it automatically runs through this data, tries to learn patterns, or tries to make predictions if you already have outcome information. For example, you want to predict cases or controls, or if you don't have any outcome information, but you just want to learn how these samples group together, what features they share, what similarities they have. So these techniques are typical machine learning methods that could be very helpful in those situations. So just to give more solid examples of practical applications of machine learning. So for example, if you have a clinical cohort of patients with gut microbiome data and they have an autoimmune disease developed, like type 1 diabetes or celiac disease, you could use machine learning to identify new biomarkers or new predictors or driving factors that will help on the early detection of these outcomes. Once you train this model on an existing cohort, then it becomes interesting, because then you can use this model, as long as it's performing well, you can leverage this model to make predictions on new data on patients that have not been diagnosed yet on a clinic or a new cohort, new independent validation study. And then you can leverage these models to help patients detecting or predicting the disease outcome before it happens, predict the disease outcome when it's going to happen, how much risk a certain patient or participant has compared to the average population that the model was trained on. So in this course, you have a portion of foundational knowledge about these machine learning models, the most useful ones in the field of biological sciences. And you also be working with some real data sets, so you can have some hands-on, some practical experience. One thing that I would like to clarify before moving forward with the course is that there are two major types of machine learning modeling that get confused a lot often, especially with this boom of large language models like ChatGPT and Claude. These terms can be a little bit confusing, so I want to clarify them first. There is predictive modeling and generative modeling, or predictive machine learning versus generative machine learning. In the predictive machine learning, which is the one we're gonna focus on this course, we use existing data to predict an outcome. So we have existing data and existing outcomes. We train a model to learn patterns associated with those outcomes, or we train the model, we expose the model to this data sample so the model can learn these patterns. And then we have a trained model that can make predictions on new samples. So we are creating these models that can predict the probability of a certain outcome, or it can group samples together by the features that they share, like do data mining, for example, or clustering. So what we are really doing here, we are identifying patterns and derive a model that is accurate enough that can be used for making new predictions. It can become like a clinically useful tool, for example. When we talk about generative modeling, which is not the focus of this course, that's when we're talking about those models that can learn existing data to create fake new data. That's not the goal of this course. And it's a completely different approach and application. In these generative models, they create new data based on existing data. They try to create new data that looks like existing data. So for example, given a text that you write on the prompt, it's going to try to create a response that would be the most similar to a real response. That's what ChatGPT would do, for example. And I'm a little bit against that in science, because in science we can't have models that hallucinate like that to predict diagnostics of patients. So generative modeling is not good for the type of analysis we are focusing on here. If we want to identify patterns in the data, predict probabilities or risk of disease, we need predictive modeling that doesn't hallucinate like that, that doesn't make fake data like that. So that's why I think it makes more sense to focus 100% on predictive modeling here. So just to summarize what I already mentioned, our focus here is on predictive modeling. We use it for regression, classification, pattern recognition, interpretation of the results, and decision making. So we are working with real biological data sets. We're not generating fake data. Machine learning and traditional statistics can also be a little bit difficult sometimes to distinguish between each other. So I just wanted to highlight a few key differences between these methods. So in terms of data handling, for example, in machine learning, we usually work with big data, hundreds or thousands or hundreds of thousands of samples. And the raw data usually needs splitting and reprocessing into training and test data, so you can train your model and you can evaluate how your model is doing. In traditional statistics, we don't apply that kind of approach. We use smaller data sets with predefined models or distributions that describe the data patterns, and we try to see if the small data sets fits into those rules. In terms of data input, machine learning will have a more data-driven type of approach. The model parameters will be defined based on the seen data. So it's very critical that the data is sampled, randomized, and transformed correctly, preprocessed correctly, so we can maximize the accuracy of the prediction of these models. The traditional statistics is slightly different. It starts with a set of rules and parameters that are predefined, describing how a certain phenomena or biological system works. And then we apply those parameters to see if the data fit to those parameters. So they go into like opposite ways, pretty much. In terms of results, in traditional statistics, you typically see a p-value, which would be the probability of a certain event or a certain condition to be observed by chance. So it's more focused towards the level of uncertainty of a certain certification or a certain classification or a certain treatment. In terms of machine learning, the probability is more like a measurement of how sure the model is about a certain prediction, right? It's more about the level of certainty. So they kind of opposite as well. In terms of assumptions as well, as I mentioned before, machine learning is very data-driven. The models will learn the rules, will learn hypotheses from the training data, from the data that is provided to the model. That's completely opposite to the traditional statistics, where we have a set of rules that are predefined, and then we see if the data fits to those rules. We try to prove or disprove that certain hypothesis that is already formed before we even see the data. And in terms of data distribution, machine learning will learn the distribution of the data. You don't need to set any rules about data distribution. You don't have to create any assumptions about data distribution. So there's more flexibility in terms of the data format and distribution compared to traditional statistics, where you would have to know the data distribution so you can apply the correct parameters, you can apply the correct statistical tests. So that would be an advantage of machine learning.